Okay, guys, so we're going to start with the first set of notes for genetics on this first page, where to begin talking about genetics, we're really going to start by talking about the father of genetics, which is Gregor Mendel. So Gregor Mendel is known as the father of genetics because he really came up with genetics, or the scientific study of heredity, which is how traits are inherited. So he was an Austrian monk, and I just think that's a cool fact because back in the 1800s, 1700s, in order for people to be a scientist or really spend like their life looking at scientific discoveries, their best option was to be a monk. Like if they didn't come from money or they weren't in the aristocracy, then they would become monks because, what are they called? The places where monks live. They were able to provide, a nice place for them to work and study science apart from their other monk duties. They could, pick up a passion project of whatever it was. So really, I just think that's a cool fact considering the time and what he was able to discover in the monastery. I googled it. Where they live is called the monastery. So in the monastery, they were really able to research or do whatever they found interesting. So that's where a lot of our scientists got their beginning, was in the monastery. So Mendel believed that traits were passed down in different patterns. So he didn't think it was so straightforward where like, oh, half of this goes here, half of this goes here, or like this trait you get from your father, whatever. He figured out that there were different patterns, and since it's a pattern, then we can predict what it's going to look like since it's a pattern. In his studies, he worked a lot with Pisum sativum, which are pea plants. He chose pea plants because these plants have several traits, not just one or two traits. They had several traits that completely exhibited one of two, not a combination, of traits, making it really easy to study. So it wasn't like if you mixed a tall plant with a short plant, they wouldn't be a medium-sized plant. If you mix a tall plant with a short plant, they would either be tall or short, depending on the inheritance pattern. So those were kind of the things he was looking at. It wasn't a combination of both of the traits. It was one or the other, kind of how we looked at if you have freckles or if you don't. Those traits that are either this or that. Some of these traits that he looked at was seed shape. So the shape of the pea seeds, they could be round or wrinkled. They wouldn't be like, kind of wrinkly, like they are either round or they're wrinkled. He looked at seed color, yellow or green. He looked at flower color, which was purple or white. So notice how I have or on all of these, because it wasn't like, maybe a little and this one, like it was either or. Where their flowers were was another trait. Plant height was another trait. And pod shape. Oh, and pod color. So all of those different things he was able to cross or breed to figure out how they were inherited, if it was either this trait or that trait. And as we will find out later, I'll talk about it down in the next paragraph. So Another important fact that I like to include on Mendel, it wasn't like he spent some time working on this. This experiment took him 8 years of his life, and he grew over 10,000 pea plants. So it wasn't like just a little quick, easy project. Like, this was his life's work, and he was dedicated to it. So it took him over 8 years and over 10,000 pea plants to test his hypothesis. So now that you know what Mendel was kind of testing, seed shape, seed color, flower position, plant height, pod shape, pod color, because he realized that things are inherited in patterns, and that was what he was testing, I want you to write a hypothesis. You are going to write a hypothesis. So if blah da 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 da, then blah da 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 da. You're going to go ahead and write that hypothesis that fit with his experiments. So moving on down, let's talk about what Mendel found after those 8 years and 10,000 pea plants. So his conclusions, he figured out quite a few things. So the first thing he figured out was that yes, parents transmit information. Wrong thing. To their offspring through what he called factors. He didn't really have a word for it, but he realized like there's something going on here. Parents are passing this down through these factors. Air quotes. Factors. So since parents pass the information to the offspring, that means that the offspring has two factors because you get one from each parent. You get one from the male, one from the female. So each individual has two factors for a trait, one from each parent. And before he really figured out what factors he was talking about, he used letters to easily represent these factors. So since each parent can have either the dominant or the recessive, and then they give one of those to the offspring, there are different combinations of these factors that the offspring could get. So if the genes are different, then they are called heterozygous. The word heter, we know, means different, meaning that they are different genes. So a big A and a little a. We don't represent them with different letters because they're still the same gene, but just different forms of the gene, right? So it's still like for eye color, or not eye color, but for freckles, and it would either be freckles or no freckles, right? So it's still the gene for freckles, just whether or not, or which gene you have for it. And so then if the genes are the same, we call them homozygous. Homo meaning same, as we know. And this could be two big letters or two little letters together. They're the same form of the gene. The dominant gene is known, or we represent it with the big letter, is the dominant gene, and that's the homozygous dominant. So homozygous dominant, we'd call them big A, big A. This one is homozygous dominant. And then if they're both recessive or both have the little letter, then they're homozygous recessive. So you can't ever just say homozygous. You have to specify homozygous recessive or homozygous dominant. You can always just say heterozygous because that means both of them. But you can't just say homozygous. You have to say homozygous dominant or homozygous recessive to really talk about which one you're referring to. So since Mendel came up with this idea of factors, we have given it a name. And so these factors are now called alleles. Make sure you are listening to me when I pronounce that word because it's an easy word to mispronounce. But these alleles make up the following. So the alleles can tell us a few things. It can tell us the genotype, which are basically like which alleles the organism has, or the type of genes, aka big A, little a, big A, little a, all the different combinations of the genes that they could have would be their genotype, because it's their genes. And then from the alleles, you can also figure out the phenotype. And the phenotype is the way that the trait is expressed physically, aka tall, short, yellow, green. And the way that I remember this, and we'll teach y'all how to remember, is that the phenotype is represented physically. So they both start with a f, phenotype and physically. And phenotypes can kind of be subjective. So if you're talking about tall, short, yellow, green, all those things can be subjective to the viewer. Okay, so we're starting on the second page of notes here after we've completed that one. So after Mendel did all these experiments over 8 years and over 10,000 pea plants, his hypothesis, maybe what he thought was going to happen, was that these traits were inherited in patterns. And so if that's the case, if it is inherited in patterns, then if you know part of the sequence, then you can predict what's going to happen next, right? If it's a pattern, you can predict it. And so through all of his experiments, he began to predict what the outcome in the offspring was going to be. And he made some scientific laws. So we're still talking about Mendelian inheritance, basically the type of inheritance that Mendel founded. So the first thing is the law of dominance. And the law of dominance just says that the dominant allele masks the recessive allele. And this is what we were kind of referring to in the last page, the law of dominance, where if you have one dominant allele, that's the trait that's going to show, because it doesn't matter what the second one is. If it was another dominant trait or if it's a recessive trait, it's going to show the dominant allele. So only one dominant allele is required to express the trait. For example, if the genotype, that type of genes for a trait, is heterozygous. And remember, heter means different, so they're the different forms of the trait. They're different alleles. The dominant phenotype shows. So just because there's that capital A there, that dominant allele, it does not matter what the second one is, because that dominant allele is going to mask it. And then here's this common misconception that the dominant trait is not always the most common. It's a common misconception. So they think, oh, just because it's a dominant trait, it's always going to be more common. And from our experiment, even with collecting data, we found that that wasn't the case, and that the dominant trait is not always more common. It just depends on the type of genes that are in that population. And so this idea, the law of dominance, when we talk about Mendelian inheritance, we will often call it complete dominance. So if you didn't have that written already, write it. Where if you have a heterozygote with a big A and a little a, it doesn't really matter what that second allele is, because no matter what, if it was a big A or big A little a, it's going to show the dominant trait. It's not going to show an intermediate. If it's big A little a, it's not going to like show half the dominant and half of the recessive. If that dominant trait is there, it will show that trait. So that's called complete dominance. And because we will talk about other types of inheritance patterns, and so it's good to know that when we talk about Mendelian inheritance, when we're talking about complete dominance. So not only did he discover the law of dominance, he also figured out a couple more things. So he made a law of segregation. And if you think of the word segregation, like in the way that y'all are probably most familiar with it, with like civil rights terms where things were separated. So it's kind of the same thing here, where instead we're talking about genes and genetics. So members of each pair of alleles separate when gametes are formed. And gametes are just the haploid sex cell. So I'm going to write that up here. So that would be like the sperm or the egg cell that is ready to either fertilize or be fertilized to form a zygote, which is the new life, right? So, and remember, haploid means half. So it's the sex cell that has half the genetic information. But when these are formed is when only one of the alleles goes into that. So as an individual, you have two alleles, but when these gametes are formed, only one allele goes into each cell, so that it's half, so that you only get one allele from your mom and one allele from your dad. And then during meiosis, sister chromatids of the chromosome separate. So that instead of having your nice chromosome that looks like this, you have it like that, where they're each in their own little cell. And then the third law that Mendel came up with is the law of independent assortment. An independent assortment basically means that two or more pairs of alleles segregate independently. I'm going to underline that. Segregate independently of one another during gamete formation. So it's not like, oh, this allele and this allele have to stick together, and like if this allele is chosen, then this allele has to be chosen. No, it's really just random how they're lined up and how they're chosen. So if you have down here, if you have big I little I and big O little o, you could have these two outcomes, big I big O or little I little o. That's one possible outcome. And then if you still had big I and big O little o, which are both heterozygotes, you could also end up with this combination. So the sister chromatids sort independently of other chromatids. And I want you to mark this on here. Wherever you can find room, make it. But the law of segregation and the law of independent assortment really are one of the things that lead to genetic variation. I'm going to add lead to genetic variation. Because if you think about it, you're not exactly one half of your mom are exactly one half of your dad. Their traits kind of mesh. And it's because of the law of segregation and the law of independent assortment that we are this nice combination and not exactly half, like a weird collage of your parents. And so, like I was saying, Mendel knew there was this pattern. And so, if we know part of the sequence, then we can predict what would happen. So, from his experiments, he was able to figure out these patterns. He was able to calculate the probability, which is the likelihood that an event will occur, and he used it to calculate the likelihood of a trait being expressed by an individual. This likelihood, this probability, can be expressed as a ratio. So here you'd say there's a 3-to-1 ratio. It could be expressed as a fraction or 3/4, or it could be expressed as a percentage, and you could say 75%. So, just another little math thing for you. The probability of multiple events occurring is the product of the individual probabilities, meaning that the event is not affected by what last happened. For example, we can work out the probability of having 5 children all of the same sex. So to work that out, let's go down here. So the probability of having a girl your first time is 50-50 chance, roughly. So you have a 50-50 chance of having a girl the first birth. And then the second birth, the fact that you had a girl here does not affect your next outcome of sex. So the next one would also be 1/2. And then the third child still has a 50-50 chance. Fourth child still has a 50-50 chance. Fifth child still has a 50-50 chance. And so the probability of having 5 children all of the same sex would be 1/32, because you take 2 times 2 times 2 times 2 times 2, which is 32. And so a really handy way to figure out the outcome of the cross is using a Punnett square. Punnett squares are my favorite. Punnett squares are just a graphic device used to determine the probability of phenotypes and genotypes. Because if we know the genotype and we know the dominance pattern, then we can figure out the phenotype, right? If we know the genes and we know which trait is dominant, then we can figure out which trait the offspring is going to have, what they're going to look like. So the alleles of the parental generation, or the parents, are written across the top and along the side. And then the alleles are written in the grid. So I have this grid down here. We can work it out, and you should be doing this too. So let's say one of our parents is a heterozygote. And it doesn't matter if you always write the female genotype on top or on the side. It doesn't matter because it'll work out the same. And then let's say the other one was homozygous recessive. Let me do that in a different color. Okay, so you'd bring this A down and this A across so that you get big A, little a. You'd bring this A down and this A across again so that you get little a, little a. you'd bring that top A down all the way in that square. And then you'd bring this one over. And you'd bring it over there too. And then this one would come down here. So that you'd have a, little a. Okay? But here you can see in each possible outcome, one of the alleles came from this parent and the other allele came from this parent. Okay, that's why I decided to use two colors so you could really see. So here, if I were asking about, let's say, freckles, because freckles is a dominant trait. So if I were to ask, what's the probability of the offspring having freckles, you would say these two outcomes right here. So there's a 50% that they would have freckles and a 50% chance that they would not have freckles. Or you could say a 1-to-1 ratio. Or you could say 1/2. All of those things would be different ways to say the same information. Okay, so let's look at Punnett squares even more on the next page. So moving on. So there are types of crosses that we could do with genes. And the first and most common type that we'll be dealing with is a monohybrid cross. Remember, mono means one. So it's a cross that involves one trait. But remember that one trait is two alleles. So, for example, this is the one trait with the two alleles. The grid has 4 squares because there are 4 possible outcomes. And from this, you can find 2 things. You can find the genotypic ratio, and that's the number of times a genotype would appear in the offspring after a test cross. So it's a test cross, meaning that, if you know, like, the parent's genotype and they don't have children yet, you could do this cross and you could say, okay, there's a 50% chance that they would have this trait or whatever. And that could be used to guide decisions and things like that. So the way genotypic ratios are written is that the number of homozygous dominant goes first, and then you put those little dots, the colon right there, to separate the numbers. And then you have the number of heterozygous individuals. And then you have the number of homozygous recessive. And we'll do practices on these in a second. And that's for the genotypic ratio, because in a genotypic ratio, or there are 3 possible genotypes. Remember, you could have big A, you could have big A, and you could have little a, depending on the parental genotypes. But let's look at phenotypes. So here, we only have 2 possible phenotype outcomes, right? You either have the dominant trait or you have the recessive trait. There's no in between. It's either dominant or recessive. So here we only have to compare the number of dominant traits with the number of recessive traits. So it gives you this example right here where it says cross a heterozygous female and a homozygous recessive male and give the genotypic and phenotypic ratios. So like I said, it does not matter which genotype you write on top. So I'm just going to go with the heterozygote on top, using W's this time. And this is a good time to say it. If you're using letters that like look the same if they're capital or lowercase. You really have to exaggerate the littleness of the little letters. So like W's and S's and U's, I guess. I don't know. But you get what I'm saying, where they look the same. It's not like an A where they're two completely different letters, not just different sizes. So this is a heterozygous because we have a big W and a little w. And then let's do red. For a homozygous recessive, we're going to be 2 little w's. So then we do the cross and we get a big W, little w, little w, little w, big W, little w, little w, little w. Okay, so now again you can see in each square there's one from this parental genotype and one from this parental genotype. So in this cross between these two individuals, these are the possible outcomes for that one offspring. So let's do the genotypic ratio. So I'm just going to say G for genotypic ratio. The number of homozygous dominant traits, which would be the WW, big W, big W. Can't really tell that because those look small. Big W, big W. Are there any big W, big W's here? There are not. So we'd say 0 because there's 0 homozygous dominant. How many heterozygous squares are there? This one and this one, so 2. And how many homozygous recessive are there? These 2. So the genotypic ratio, you could just say 0 to 2 to 2. And if we know what numbers go where, then we know, okay, there are 0 homozygous dominant, 2 heterozygous, and 2 homozygous recessives. So let's look at the phenotypic ratio. So the number out of the cross that are going to show the dominant trait is 2. These two squares. And then the number that'll show the recessive trait is also 2. So they have a 2 to 2 phenotypic ratio. Or you could then reduce it and say 1 to 1. And so, since this is just like a cursory introduction to genetics, we will mainly be working with monohybrid crosses. But if you think about it, like these are the ones that we were doing when we collected our data. So like earlobes and freckles and dimples, those can all be worked out with a monohybrid cross. But we talked about, like, are all genes going to be that simple where they only have 2 alleles? No, it can get very complicated. So there's also dihybrid crosses. That involve 2 traits, therefore 4 alleles. So since there are two traits, there would be 2 different letters. Since there are 2 different types of traits, it would have 16 squares, and the phenotypic ratio of a dihybrid cross between 2 individuals that are heterozygous for both traits, then it gets really complicated, and you'd say there's a 9 to 3 to 3 to 1 ratio of outcomes. And then, since we will be using Punnett squares a lot, I wanted to talk about this extremely common misconception because I don't want y'all to make this mistake. So, lots of times if we're like looking at our Punnett square, it's easy to think like, oh, this is one offspring, and this is another offspring, and this is another offspring, and this is another offspring. So, lots of times people say like, oh, 1 out of the 4 kids would have this trait. But that's not what this is doing at all. This Punnett squares are really for one offspring to see the possible outcomes of this one offspring. And then if the parents reproduce again, it'll be the same outcome because their genes haven't changed, So using a Punnett square shows all of the possible outcomes. All of the possible outcomes for one offspring. And I'm going to underline, circle one offspring because that's super important. It's not saying like 1 out of their 4 kids would have this. No, in one kid, there is however percent chance of inheriting a certain trait. So in a monohybrid cross, the Punnett square shows the 4 possible combinations of inherited alleles for one offspring. Again, gonna underline that, circle it, because that's super important. Okay, so that kind of clears it up for Mendelian inheritance. And like I mentioned before, there are other types of inheritance that we are going to mention and go over a lot. So, there are a few non-Mendelian inheritances. The first that we're going to talk about are autosomal traits. And autosomal traits occur on autosomal chromosomes. Which are the 22 pairs of chromosomes that are not your sex cells. So basically, autosome means not sex cells. And since it just occurs on like the things that code for literally everything else in your body that has nothing to do with the sex chromosomes, it'll show up in both sexes equally. So it doesn't matter if you're male or female, whichever, it will affect at the same rate because it has nothing to do with the X or Y chromosomes. However, there are plenty of disorders or traits that we're going to talk about that are sex-linked. And those traits occur on the Y or X chromosome. And it usually only affects males. And we'll talk more about that a lot in class together. But when we talk about sex-linked, we're mainly going to be talking about X-linked traits. Because the Y chromosome doesn't really code for a lot. It's mainly the one X chromosome that does everything out of your sex chromosomes. The Y chromosome's like really small, kind of dinky, and really doesn't do much. So when we talk about sex-linked traits, all of these sex-linked traits that we're going to mention are on the X chromosome. There are some that are on the Y, but we won't mention any. So another type of inheritance is incomplete dominance. And in incomplete dominance, the trait is intermediate, which means like it's mixed. That's in the middle of the two traits, okay? So, for example, if we have red snapdragons, which is a type of flower, and we have white snapdragons, let's say in complete dominance, you can only have red or white. But in incomplete dominance, where there could be an intermediate, you could have red and white. And if you mix those two together, what color are you going to get? You're going to get pink. So red or pink is the intermediate of the red and white colors. And because of this intermediate, it means that in incomplete dominance, an offspring can have one of 3 possible phenotypes. So there are still only 3 genotypes, but now there are 3 phenotypes as well. And for incomplete dominance, we will still have, like, let's say, big F, big F. So this one would be red. And then if you had big F, this one would be pink. And then if you had little f, this one would be white. Okay, so now the heterozygote is at the intermediate. Okay, the next type of dominance that we'll talk about is codominance. And in heterozygous individuals, both traits are expressed, meaning that they share dominance. So if you think about like co-workers, they have to work together. If you think about codominance, the traits kind of have to work together. So, for example, in horses, if there's a red coat and a white coat, then they'll get this roan or red/white coat. So it obviously won't be a pink horse. It won't just be a red horse. It won't just be a white horse. It'll be red and white. It'll be splotches. And a really common example is in certain types of chickens. They can have a white feathered and a black feathered chicken cross, and then they get this spotted chicken. So if it was complete dominance, then it would be either a white or black chicken. In incomplete dominance, it would be like this gray chicken, but in codominance, since they work together, it's black and white. Okay? And with multiple alleles, more than 2 alleles are possible, but an individual only has 2 alleles. So there's a lot of combinations that could happen. And the most common type of example of codominance is blood type, where you can have the A, you can have the B, or you can not have one at all. So like if you have A positive or A negative, you have that genotype. If you have B positive or B negative, B minus, then you have that. And then there's the chance that you don't have either of those alleles and you have O blood, type O plus or minus blood. And that's when you'd have that genotype. And when we do codominance, I do want you writing this part in if you haven't written anything else. In codominance, let's go with this example right here. So say we had a white feathered and then a black chicken. In codominance, we do use different letters. So here you'd have black and white, black and white, black and white, and black and white. There's a 100% chance that it would be black and white if you had this genotype and this genotype. But we use different letters, different alleles, since they can both be expressed. And so kind of everything that we've talked about so far, those have been the really simple alleles. So in like different organisms, they might have more alleles, or they might have more traits that only require a few alleles. So like the traits like freckles and dimples and hairlines and crossing your hands, those things are simpler traits. And that's why we use them as examples. But most common in humans and most other complex organisms, like mammals. In humans, though, since we know ourselves best and we can use this as a best example, most of our traits are polygenic. So if you think of what poly means, it's where traits are controlled by more than one gene. Because poly means many, and genic means genes. So polygenic is many genes. So instead of having like a big A, little a, it would just be hundreds of alleles long. And also polygenic traits can be influenced by environmental factors. And these environmental factors aren't just like what you're thinking about, like with the environment. They're also just like things that aren't already coded for in your genes. So if you think of hormones, depending on the amount of hormones they have, like if you have a hormone imbalance, it could affect a lot of these traits, these polygenic traits. If any drugs are related, if chemicals, light, if you think about like skin color, for example, it's on such a wide spectrum because it's a polygenic trait, and it can also be influenced by the environment. So like you're super tan in the summer, or you're paler in the winter, or you stay pretty much pale all throughout the year, or your skin tone stays constant, whatever. That's a example of a environmental factor. And then also nutrition obviously affects like weight, but it can also affect height a lot. Like if your nutrition is better, in your early on years, then you'll have a greater height advantage. But if like your parents are both 5 feet tall and you like eat good and healthy when you're younger, there's a pretty good chance that you won't be 6 feet tall. Because it still has a genetic component, but also has an environmental component too. So, like we've mentioned, height, weight, skin, hair color, and pigmentation of fur. Like in us, our hair on the rest of our body, or just fur in other animals, other mammals. So, if we're collecting all of these traits, it can get pretty complicated because there's so many. And in order to figure out our traits, we not only need to know our phenotype, but it helps us if we know our parents' phenotypes and genotypes. That way maybe we can figure out our specific genotype. So if you think about like the traits that you have that are the dominant traits, like on that paper, we know for sure that you have at least one of the dominant alleles, but we don't know about the second allele. That's why I had you leave it blank, because it could be another dominant allele, it could be a recessive allele. You kind of have to look at your parents and maybe your siblings if you have some, and piece it together. It's like a puzzle. And so one of the easiest ways to like represent that puzzle is through a pedigree. And pedigrees are just a graphic device showing occurrences of genetic traits over several generations. Because if, like Mendel said, these traits are inherited through patterns, it's easy to see patterns if you look at a big population over a long period of time. Easy to see patterns. So it's basically, if you think about it, a family tree showing traits in the form of phenotypes, and from this, genotypes can be derived. So like I said, if you know a phenotype and you know the dominance pattern, then you can figure out the genotype from that. So like if I can't roll my tongue, then I know that my genotype, if rolling tongue is dominant, this would be a rolling, it could be a big R or a little r. That would mean you can roll your tongue. But if I was not a roller, then I know for sure that that is my genotype. Okay, so if only the phenotypes are known, it's easier to work out the genotypes, because not all of us obviously know our genotypes off of the top of our head because we've never had our genome sequenced. But using our phenotypes and the dominance inheritance, we can figure out what our genotypes are. It's pretty fun. And so like I've already told y'all, if it's only known that the dominant gene is possessed, leave the second allele as a blank, like I did here and like you did on your worksheets. Okay, and this is just like the basic notes for all of these. We will be doing worksheets. We'll be talking about it in class. We'll be doing projects with all these things so that we can understand more about genetics.